methods, molecular methods, and serological methods. Here I have enlisted different methods. When you take veneer physical methods, that is infection by dry seep, soaking method, the suspension of wash method, microscopic methods, and by using indicator was that what we can call bioassay or biochemical test. Then different incubation methods, blotter method, 2,4-D method, freezing method. Then the advanced methods like molecular methods and some serological methods. So in 19 years, before 100 years ago, Intner and Doyer, they developed different just incubation methods, infection methods. They are at present after ISTA, ISTA have 19 communities. Out of 19 communities, the plant disease committee, also called seed health committee, is very, very important. That committee decides the protocol for seed health testing, and they will give the different tests, enlist the different tests for the different pathogens. So it has established in 1957, whatever the incubation methods or physical methods, they are outdated, and we should look for new advanced methods and other methods. So currently, in seed testing, we are since 1935 years we are using the evaporating method, blotter test method, the bioassay, and by microscopic observation, some serological methods, not ELISA, some other agglutination test, immunoprecipitation test, other tests we were using since 35 years, but these methods are multi-stage. They are more they, that consuming more time. They are labor incentive and subjective. In this regard, the seed testing undergoes significant changes since 1957 as ISTA established seed health committee. Since then, it has revolutionized, undergone significant changes. At the same time, the merely at the, that means 1957, the only depends on incubation, bioassay, and some serological methods. Now, there is a time to advance the techniques with respect to seed health test. There are so many advanced techniques are there. They are giving accurate result and they are very feasible in the detection of seed-borne pathogens. Then even the different countries they have developed different methods. Such those methods are one is serological methods. It includes enzyme-linked immunosorbent assay, DBA, that is dot on DBA immunobinding assay, tissue print immunoassay, or we can also call that tissue print immunoblotting, lateral flow strip method. These are all the variants of this ELISA. Nowadays, especially for bacteria and the identification of viral pathogens present in the seed, they are using. Similarly, we have molecular methods. Here, molecular methods, we are primarily using specific primers for identification of seed-borne pathogens. We can use for all pathogens, all category of pathogens, bacteria, fungi, viruses, viroids, nematodes, and so on. These molecular methods includes that the PCR-based methods also there, non-PCR-based methods also there. The PCR-based methods are RT-PCR, that is reverse transcription, PCR, that is reverse by using a reverse transcription enzyme. The another one is real-time PCR, even reverse transcripted real-time PCR also is there. Bio PCR is there. The variant of this by combining serological and molecular method of PCR is there. That is immunocapture PCR. Even in that also we can see another variant, immunocapture RT-PCR. We have multiplex PCR. We have so many variants of PCR for the identification for different seed-borne pathogens. Similarly, we have the advanced technique that is based on hybridization. That is one is microarray. That we can also call it DNA chip technology. The other advanced techniques nowadays, they are majority they are using loop-mediated isothermal amplification. It doesn't need any PCR condition. Just it needs constant temperature for 60 or 65 minutes. Then next one is next generation sequencing. We can use the metagenomics data and we can sequence, we can identify unknown pathogen also by using the next generation sequence technology. These are the advanced techniques we can adapt or employed in the identification of seed-borne pathogen that ultimately helps in the seed health management. So we'll go by one. First, you take enzyme-linked immunosurveillance assay. The mainly it's based on the reaction between antigen and antibody. Once the antibody antigen reacts, there will be a change in color because there is a presence of substance in the polystyrene plate. For conducting ELISA, we need polystyrene plate. Okay. Then NSI was at least one antibody specific to particular pathogen. If you take for example, you take TME, tobacco mosaic virus. So we should add specific. The antibody should be specific to TME in polystyrene plant. I think you know what is antigen, antibody, and what is the enzyme conflict. This is the workflow of the ELISA, that's the enzyme immunosorbent assay. In this particular, what we will do, the anti-serum specific are given, as I mentioned, if you develop anti-serum specific to TME, so just you go to the plate, as I mentioned, this is antibody specific to, for example, TME. Then wash it, then you had antigen. So then again you had that antibody should combine with the enzyme. So if it is specific virus is there that particular to antibody, there will be a reaction will happen between the substrate by the reaction by the help of enzyme. If it is the virus is there will be a change in color. The intensity of color also depends on the concentration of virus particle present in that particular sample. This concentration we can analyze by using ELISA reader. So the major advantages of this method includes it is reasonably sensitive. It's not ultra sensitive, but it's reasonably sensitive. It is less susceptible to false positive because we are adding specific to the specific antibody that particular to the specific pathogen. Then this ELISA is we can only recommend for the 99% cases we can recommend for viruses. In few cases also we can recommend for bacteria. It is not good for viral or fungi. And it is a low cost. For example, if we take we can low cost, we can at a time we can handle number of samples, large samples we can handle, and we can that reading and everything it should be automation. You can get a detection kit to analyze the sample by using this method. So next method is dark immunobinding assay. It is the one of the variants of ELISA. So we are using the detection of it is also very good for identification of viral pathogens in the seeds and the plants. So if you, here the main difference between this dark immunobinding assay and ELISA. So there we are using to bind antibodies. We are using our antigen. We are using polystyrene plate. Instead of polystyrene plate, we are using the nitrocellulose membrane as a plate. So if you add antibody and antigen, the reaction will be same as mentioned in the ELISA. You see, this is nitrocellulose membrane. You add antigen of a particular pathogen protein or anything we can that can act as a antigen. Then you have antibody which linked with some conjugate enzyme. And the presence of particular pathogen is there that is specific to antibody. There will be a change in color. That indicates the presence of the particular pathogen. If there is no change in color, we can tell it's a negative result. The main advantage of this debug is a few microliters of extra samples is sufficient to detect the presence or absence of the pathogen. As I mentioned, the debug advantage over conventional ELISA. It requires only crude specific antiserum to each of the virus or bacteria and a single enzyme conjugate. The variant of ELISA is tissue print immunoassay or tissue print immunoblotting. Here it is similar, as I mentioned, dot immunoblot assay. It is similar to debug, except it does not involve tissue extraction. There is no need to extract the inoculum from the source. Just you take any lead sample, infected lead sample, you just make it disk and press it on cellulose membrane, which is already having specific antibody. If it is the sample is infected with any particular pathogen, and if it is specific antibody on nitrous cellulose membrane, you can get positive result. If not, you will get negative result. It is it's a we can tell it's a variant of DIPA. The main the advantage is eliminating the extraction step. So in ELISA we can do extraction and purification and everything. In case of DIBA, we should need extraction. So there is no need of this polystyrene plate. Just on nitrocellulose, if you fix it, you can get the result. Here there is no need extraction, but we need nitrocellulose membrane. The another variant of LSI is lateral flow state method. I think this is as same as I think we all know about pregnancy test kit. Like that only. Just you see this picture. So the lateral flow state, as I mentioned, used for detecting viruses, bacteria, is a variation of ELISA. The antibodies of the particular pathogen, they are immobile and also test strip. Okay, the test does not require any major equipment. Just is like you keep the lateral flow are suitable for on-site use. If you want to test on-field conditions, you can use this method, especially for viruses. For example, you had analyzed sample here. You should have the conjugate. Then it flow down. So if it is positive, you can get the red color in control line as well as test line. If it is negative, only you get that color indication in control line. It is one of the qualitative estimation under field conditions. Now you come to that molecular methods. One of the molecular methods is well-known molecular methods is polymerase chain reaction. It is a rapid and highly specific in vitro amplification of selected DNA sequences. It's only meant for DNA sequences, selected DNA sequences for which primers can be used. As I mentioned, the suppose the pathogen or any, if it is latent infection or if it is not, we are seen on the surface of the seed on anything, if it is internally seed borne, if it is not from incubation method, in such methods we can easily use this method for the identification of seed-borne pathogens. It is highly sensitive and specific. You can get the result within 24 or 48 hours. The technique is applicable for wide range of pathogens. That means you can apply this technique for the identification bacteria, viruses, fungi, nematodes, and so on. But when you take serological methods, that only confined to the 99% confined to the viruses. In one or two cases, we can identify bacterial pathogens also. And another most important thing of this PCR, we can identify closely related species. We can easily differentiate closely related species. Even and another important advantage is even at ultra low levels in picomoles also, if the pathogen concentration present means nucleic acid present in picomoles, we can also identify the seed-borne pathogens. These are the workflow of PCR. The PCR should contain different components. One is DNA sample, that is sample from pathogens. Then you should have specific primers to the particular pathogen, and you should have oligonucleotide, Taq polymerase mixer, and so on. This is the workflow of the PCR reaction. However, it has one major disadvantage. So when we go for this technology, we should have well characterized sequences for particular pathogens. Then only we can apply. If it is not known the sequence information, it is difficult to identify from this method. The another method is reverse transcriptase PCR. It is a variant of PCR. See, the PCR is only to amplify specific DNA sequence of particular pathogen DNA sequences. So reverse transcriptase, why is for suppose if you take RNA samples, RNA viruses, we can't directly amplify with PCR. Before that, we should convert that RNA into complementary DNA, then only we can further proceed for polymerase chain reaction. As I mentioned, combining the reverse transcription of RNA into DNA, that is complementary DNA, the amplification of DNA targets using PCR reaction. Here, the main component is reverse transcriptase enzyme. If you use this reverse transcriptase enzyme, it converts RNA to complementary DNA. Then further we can proceed for PCR. So we are in this, we have two one-step RT-PCR and two-step RT-PCR. The one-step RT-PCR only gives the confirmation of RNA, then the pathogen. Then two-step RT-PCR, here we can confirm the concentration of the RNA and as well as we can identify. It is also, we can also, this method we can also call as real-time RT-PCR, that is reverse transcriptase PCR. You come to real-time or quantitative PCR. Here it is also one of the variant of the, we can call it as RT-PCR. Here also we are using the template is RNA, then we can convert into cDNA. See here in this we are using fluorescent emitted during the reaction. In normal PCR, just we are using primers. Once they amplify, we'll visualize through electrophoresis. But here there is no need to visualize through electrophoresis. Just if we add the fluorescent emitted fluorescence, some probes which are some probes with along with the specific primers. At the end of the reaction, there is the emission of fluorescence. That fluorescence we can record in the real time, at the real time on the monitor. The peak upregulation of the fluorescence and that indicates the amplification of particular pathogen DNA. As I mentioned, real-time PCR quantification eliminates post-PCR processing of PCR products. Usually in PCR, what we'll do, once the amplification has happened, we'll go with electrophoretic separation of the amplified DNA. In this case, there is no need for those steps. This helps to increase the throughput and reduce the chances of carryover contamination, and that is non-specific amplification. In PCR, there will be a chance for the amplification of non-targeted region. For example, if you use ITS primers, it will amplify non-targeted regions also. During that time, we will get the wrong results. In such cases, if you use this real-time PCR, we can avoid those non-specific amplification. So even this real-time PCR also we can amplify. Now it is ultra rapid cycling. Within 32 minutes to 2 hours, we can get the results. So less amount of RNA we needed when compared to the conventional methods. That is the flow chart of the how this real-time PCR works. How at the end of the reaction, as I mentioned, there is a emission of fluorescence. That fluorescence recorded by the monitor when the amplification happens. Here I mentioned the fluorescence is created by adding these product that is SVABR green, TACMA molecular biopons or scorpion probes. The probes are also a short nucleotide sequence, but which is labeled with some dyes or they are readily labeled. The another one is it's a method. It's also a variant of PCR. It's combination of some serology and molecular method. Here in initial step itself, while preparing this sample, the PCR tubes pre-coated with antiserum that's specific to particular pathogen. So what it increases the sensitivity more than PCR. So that's why nowadays, for the detection of, especially for RNA pathogens, RNA viruses, this method is getting popularized. Another molecular method is it's hybridization method, nucleic acid hybridization method, what we can call microarrays technology. Array means line. It's a spots. Dr. Dhananjay, Dr. Dhananjay. Sorry, we have some technical problems. Just wait for a few
Hello,
minutes.
sir. Manjunatha, sir.
Ah,
Am I audible?
sudden
Am I
off
audible, sir?
a sudden off ಆಯ್ತು just for 2 minutes. And again I say slides. Sorry. Uh, yeah, as I mentioned, the array microarray technology. Array means lines, spotted dots is arranged in lines. Each spot contains a specific DNA nucleotide DNA sequence, that nucleotide sequence that specific for particular pathogens. So DNA capture codes for each of the genes or pathogens to be detected or immunized with solid support in a spatially separated and individually addressable fashion like this. The different genes or different DNA we should arrange in different spots. So in this method, we can identify the more than two pathogens or more than two genes at a time. So nucleic acid from the sample to be tested is extracted and labeled, and this labeled nucleic acid is then uploaded to the array. For example, you take any DNA sample from the seeds and you label it and label. You just imprint on these arrays. If it is where there is a specific DNA sequence complemented to the our template, there will be a changing. There will be a hybridization will happen. There will be a changing color. Like this, you see, these are all negative results. These are all positive results. Like this, the DNA array works. The arrays can say that hybridization can be identified and the presence of the gene, pathogen or insect is resolved by the predefined position of the DNA capture for the array. This is the illustration. For example, this is hybridization site is having complementary to the unknown samples. Once it has hybridized, there will be a change in color. That color we can read by that DNA microarray analyzers. So that's why we can identify the seed-borne pathogen. This is also useful for a wide range of pathogens. The major advantages of these microarrays is high throughput. At a time, you can use bulk number of samples. That means you can screen for multiple viruses, bacteria, fungi simultaneously, provided the relevant sequence information is available in that DNA or microarrays. Combined detection, identification, and quantification of large number of fungi, bacteria, viruses, nematodes, insects present in the single ulcer. As I mentioned, is I throughout hundreds of tests could be run simultaneously, and it is very cost effective. The another method, it's recently since decade, which is getting popularized, that is loop mediated isothermal amplification. It's a novel technique that doesn't require any that is PCR amplification. Just it needs enzyme one enzyme that DST polymerase. It needs DST polymerase. It needs so based on the strand displacement activity, it will amplify the sample template DNA under constant temperature. That means isothermal conditions. It is also got higher specificity compared to PCR because it uses four primers, recognize six distinct regions of the targeted genome of a particular pathogen or any organism. Since I think few years they successfully employed in the identification of different plant viruses and the seed-borne viruses. So this is the flow chart of lab method. Here, for example, you take sample, seed sample for the processing, you extract the DNA. So you add the template into the lamp reaction mixture. So you keep it for isotherm at the constant temperature, that is 65 degrees for 16 minutes. Then later on, inactivate that PST polymerase 80 degrees for 10 minutes. Then we can observe whatever the amplification happen in the lamp due to lamp reaction. We can observe by three different methods by hardening that SYBR green light. This is then if you observe under UV spectrophotor or UV photometer, no, you can get luminescence. It is the presence of a particular pathogen. If it is absence of fluorescence, that means that is negative results. Even you can precipitate that product by using magnesium pyrophosphate. The precipitation, if precipitation happens, that indicates the presence of the targeted pathogen. Even we can visualize the cell to value pattern. The another recent since one decade, it has revolutionized next generation sequencing technology. So here main is we can extract total DNA content of the sample. This is sample, and whatever the nucleic acid will come, you can analyze through different bioinformatic tools. The major advantage here is compared to other methods, this approach does not require prior knowledge of pathogens, and even can potentially, it's very effective in identifying both known as well as unknown pathogens. That is, new pathogens also we can identify by using this new generation sequence technology. This is the flow chart of NGS, new generation technology. So you prepare this DNA sample. After preparing DNA sample, we should make library of that particular DNA sample. Then once we make library, and here in next generation sequencing, we are having different platforms. Okay, if we are having Illumina, biosequencing, TypeBio, then ion PCR, so many, and based on hydrogen concentration ion, we can sequencing nanocode is there. By using these different platforms as well as different bioinformatics tools, we can identify even unknown or new pathogens that present in the seed plant samples. Here we just need a nanogram of sample for the identification of pathogens. So what we can conclude from these advanced techniques, summary is, you see we have different serological and molecular and hybridization methods. The most of the methods, especially this DNA hybridization, loop mediated, LPS dependent NGS, they are used for the identification of the bacteria, virus, as well as viral and other pathogens also. And the almost all serological and molecular methods easily you can employ for the identification of virus. The almost all methods you can easily use for the identification of virus present in the seed sample. Some methods only specific for particular pathogens. You just see the tissue blotting immunoassay is only meant for virus. And you can see this one, reverse transcriptate viruses only, not for viral or bacteria. Like this one, it's having one advantage and disadvantages. So another method, it's not accepted by STAN, but it's recently they are doing work on it. The one multispectral image system, but on the use of computer vision. But based on this different light wavelength of lights, we can capture different images of healthy sample as well as disease sample. So when we process those images using different softwares, you can get different images. You can easily differentiate, easily discriminate the healthy and infected samples. So one example I will give. So you see that research and its co-workers in 2016, they used the multispectral image system and at 19 wavelength bands to differentiate the healthy seeds of wheat from infected seeds of wheat that is infected by Alternaria and Fusarium species. You see, you can get different images. The healthy image is totally different when compared to the wheat seed infected with Alternaria and Fusarium. So like this also we can identify and the seed-borne pathogens by using multispectral imaging system as with the help of computer vision and different statistical softwares. So what are the take-home messages from these advanced seed techniques? The one is besides unique advantage offered by these various seed and plant disease detection methods, each method has its own limitations as I mentioned in the summary. So some techniques only specify particular pathogens, some we can apply for the all type of pathogens. The another one is these assets. It is critical to rigorously evaluate their applicability, precision and accuracy in real world, high throughput testing of naturally infested seeds. The another one is to ensure that these assay work, they must be validated in stringent multi-laboratory tests which evaluate their reproducibility and repeatability. This reproducibility and repeatability this is very important when you particular method is under register under ISTA. So this is the some of the advanced technology for the identification of seed-borne pathogen. That means for the seed health. Even some premier organization association they're working with this seed health. The one is these are the premier organization. They day to day they will develop different protocols. They share the different protocols for the identification of different pathogens. They index in the pathogens that means seed health. One is International Seed Testing Association. The another one is Association of Official Seed Analysts, especially for vegetables. Then International Seed Health Institute is also for vegetables. Then National Seed Health System, which is in USA. Then another one is ICR, Indian Institute of Seed Science. It is present in India. One by one we will go. One is International Seed Testing Association. As I mentioned, here in International ISTA, they are having 19 subject focused technical committee. In that, Seed Health Committee, which is one of the ISTA technical committee, is responsible for develop and establish seed health testing method. And there are 30 different testing methods till now in ISTA rules. So whatever you develop, they need 4 points to adopt those methods. One is high specificity and sensitivity for the pathogen. The second one is simplicity and quick, cost effective. As I mentioned, it should be repeatability and reproducibility. The another one is Association of Official Seed Analysts. It's a members of different countries, that is all the members laboratory of association officials in analysis. They do not administer seed certification or GEMPA, any other crop science. All seed certification activities are administered by individual agency who are members of this ASAM, the association of. This is mainly focus on the grasses, vegetables, fruits, vegetables. They mainly focus on the seed of these crops. And another one is International Seed Health Initiative. At presently, that finance and administrative International Seed Federation coming under International Seed Federation. So as I mentioned, it started as an inventory of the vegetable seed industry in Dutch and French seed companies for the Dutch and seed companies in 1994 to monitor seed health. This organization also involves developing, evaluating, disseminating the information on the seed health testing methods. When it is started, it's only confined to the vegetables. Later on, it has started with herbage and field crops. That is in 1997, it started with herbage to develop protocols. In 1998, it taken field crops for developing the seed health standard methods. The United States, Japan, and Israel, they are coming under this initiative. That means they represent more than 75% of the world vegetable seed supply. Another one is International Seed Initiative. Sorry, this is National Seed Health System. It's a program authorized by USA, government of USA. It is meant for to give accreditation to the private and public players. to to issue the federal phytosanitary certificate based on the seed health. As I mentioned, NSCS has approved several seed health testing methods in various crops: beans, peas, some of the vegetables, brassica, carrot, cucumber, celery, spinach, corn, and so on for different seed-borne pathogen, fungi, bacteria, and viruses. Is the another one is Indian Council for Agriculture Research, that is Indian Institute of Seed Science. This organization also they work on detection and diagnosis of seed-borne pathogens, and even they also work on the management of seed health management and testing. What is the common outcome from these organizations with respect to seed health? That means they should achieve the protocols and procedure of seed health testing by these carefully achieving the uniform international standard. Whatever they will develop, they should be uniform throughout the world, and should have uniform international seed standards with respect to seed health testing and seed pathology. That will make easy for the seed trade and quarantine purpose. The conclusion and future focus uh, of the seed health: detection and diagnosis of pathogen critical for, as I mentioned, it's a very critical for the seed trade and germplasm exchange. And what is the future? There is a necessary for web-based information protocol sharing time. Here, the different countries they develop different protocols. There is a common platform for sharing these diagnostic protocols that is on web-based. That database and experts use it for quarantine purpose. Then molecular techniques for detection of pathogens in quarantine assume a great significance, as I mentioned, by looking at their reproducibility and repeatability, but it need to be improved for high throughput and user friendliness. All quarantine laboratories need to have anti-sera for the all plant viruses, which facilitate the interaction of the exotic viruses. It is the must and take home message that whatever the quarantine stations present in India should have the regular anti-sera for the domestic quarantine virus as well as foreign international quarantine viruses. And another important thing is there is a need to establish national diagnostic network of plant and pest. Once we develop network, network should include anti-sera bank, database of primers, because anti-sera is for serological test, database of primers through molecular methods easily you can identify, seed of indicator host. It is also for biosec test we can do seeds of the different crops, for example amaranth, so many are there for the. Then national DNA bank of plant pests we should have. Lateral flow strip or dipsticks which can detect multiple pests, multiple PCR, real-time PCR, and loop meditate microarray etc. we should have at the quarantine centers. And another feature for the way forward is a national biosecurity chip for diagnosis of all current threats to crop plant would be backed on for strengthening the program of plant quarantine. That means if you make seed health is strong, we should need to develop some diagnostic methods, that is national biosecurity chip. Then another one is, as I mentioned, national diagnostic network for plant test. Suppose if it is established, can be storage of information of biology of pest, detection of protocols and policies. If you develop these things, no, it will help easy movement of seeds from one country to another country without any bias. Also, regional working groups of experts for detection and identification of plant pests does need to be formed to share expertise facilities. For example, in South Asia, where the borders are contiguous, this would be helpful in avoiding the introduction of pests, unknown pests in the region, also the transboundary movement of pests within the South Asian region. Thank you. Thank you so much. If you have any doubts, ask.
Thank you so much, sir, for your nice presentation on advances in seed health testing methods in general. With the permission of speaker, the session is open for discussion. If any questions are there from participant side, means just unmute yourself and you can ask.